let's continue discussing replication of influenza virus. We know that not all, majority of RNA viruses replicate in cytoplasm. However, influenza virus is an exception. It does replicate in nucleus. And look on this nucleocapsid, where RNA is wrapped around the NP protein, and this helical conformation has a central loop and is associated with RNA-dependent RNA polymerase, which is trimeric, formed by PB1, PB2, and PA. And these nucleoproteins have NLS, nuclear localization signal, and enables this nucleocapsid to get into the nucleus by interacting with importin alpha. Now, once a viral nucleocapsid gets into nucleus, the polymerase start to synthesize mRNA. However, for synthesis of naked mRNA, RNA polymerase required primer, and that primer is snatched from the cellular mRNAs. You remember, PB2 binds to cellular mRNA 5' cap. PB1 cleaves it, and this polymerase uses that cap from cellular mRNA as a primer to synthesize a naked mRNA. Those mRNAs are shuffled in cytoplasm, where they are translated into the proteins, which are afterwards transported back into the nucleus because they have also NLS signals. Once enough of nucleoprotein, that capsid protein, is made, this switches polymerase synthesis from mRNA to antigenome, and from antigenome is afterwards replicated to genome. And this can only happen if there is a sufficient amount of nucleoprotein. And it is because you remember that RNA minus viruses have always their RNA packaged with capsid, nucleocapsid. And once the switch happens and polymerase start to synthesize antigenome and genome, it does not require primer. So cap snatching is only necessary for synthesis of mRNA. And here it is how it happens. So PB2 binds to cellular 5' cap. It brings PB1 there, which cleaves it, and that cap is afterwards used as a primer for synthesis of mRNA, cap snatching. So replication of influenza virus depends on the synthesis of cellular mRNA. So if we block cellular mRNA synthesis by actinomycin D or amanitin, so it will also block replication of the virus because the synthesis of mRNA, viral mRNA, will be blocked. Now, once that capsid protein, that nucleoprotein, is synthesized, and there is enough of those, basically polymerase switches from synthesis of mRNA to synthesis of antigenome, and from antigenome to genome. So influenza A mRNAs undergo alternative splicing only for a 2 segment, you remember. M1 and S1 and M2 and S2. So this replication, I stress again, replication and synthesis of antigenome and genome does not require primers. Yes. Now, once the nucleocapsid are synthesized and they are associated with polymerase, they are transported into cytoplasm. I want to stress importance of NS1 proteins, the non-structural protein 1. Not only it does interfere with many antiviral pathways, but it also interferes with polyadenylation of the cellular mRNA. So it binds to cellular mRNA and blocks the function of polyadenylation factors. So think about that. Influenza snatches cap from mRNA, cellular mRNA, so makes them non-functional. And it also blocks polyadenylation, so it prevents synthesis of mRNA for cellular proteins. In addition to that, PB1F2 also can be translocated into mitochondrial membrane and trigger apoptosis. So these are two factors with another contributing to cell death of the ciliated epithelium, which is a beginning or which is a base for those symptoms which follow. Now let's go back to NS protein. It does suppresses antiviral pathway. You recall RIG1, cytoplasmic PRR sensor, which recognizes viral RNA. So NS1 protein, it inhibits its activity. It also inhibits PKR and 2' 5' oligosyntase. Both are protein triggered by interferon, and it activates PI3 kinase. So influenza virus has very high impact on the survival of the cells which are infected. Now, let's go back to replication. Once those envelope protein, neuraminidase and hemagglutinin are synthesized, they are targeted from ER and Golgi to cell membranes when they are inserted, and they attract matrix protein. And the matrix protein would attract those nucleocapsid, 8 of them. And basically, virus start to assemble and start to buds from cell. However, remember, on the surface, there is a lot of those sugars which have sialic acid, and hemagglutinin, which is inserted, would interact with them and hold that viral particle on the cell membrane. Here, neuraminidase comes, it cleaves that sialic acid and releases a viral particle. So hemagglutinin is important for entry of the virus. Neuraminidase is important for release of viral particle from cell. And let's go back to hemagglutinin. You recall that hemagglutinin has to be cleaved to yield that hemagglutinin 1, which has a binding domain for sialic acid, and hemagglutinin 2, which has fusion protein. Now, most of the viral strain, influenza strain, are limited to respiratory tract, so they can infect our upper and lower respiratory tract, and it is because this cleavage is mediated by serine proteases, which are only present on respiratory tract. However, under certain circumstances, new viruses can adapt. This cleavage site is mutated and acquire capability to be cleaved by furin. And furin is present in many tissue and organs. As a result, when this happened, and it happened particularly in the birds influenza, afterwards the virus can replicate to high level in any organs, whether it is brain, lung, spleen, liver. And this causes high pathogenicity. So that cleavage site is important for limiting influenza replication in the respiratory tract. So limitation, variability, epidemic, pandemic, how those changes, those antigenic changes happen. There are two major mechanisms, antigenic drift and antigenic shift. Antigenic drift is very slow, continuous accumulation of point mutations. We know that RNA-dependent RNA polymerase doesn't have proofreading, very good proofreading capability, so it continuously introduces point mutation. And this point mutation can cause minor changes in hemagglutinin and neuraminidase. But until those changes are minor, our antibody can cross-react to it. They are not 100% efficient, but they still can cause some preventative measures, so they have cross-reactivity. However, if two different viruses, let's say bird virus and human virus, infect the same cell, and those segments, the nucleocapsid, reassort, they can create so-called antigenic shift, the virus with completely new characteristics. So here it is in detail. Antigenic drift on left, you can see those point mutation can change basically certain sequences on hemagglutinin neuraminidase, but there still can be cross-reactivity with our already produced antibodies, and there is partial protection. However, if birds, for example, influenza virus and human influenza infect pigs, you recall birds use 2,3 sialic acid to enter, while human influenza uses 2,6 sialic acid as a receptor. In pigs, you can find both 2,3 and 2,6. Therefore, in pig can be infected by bird influenza virus, by human influenza virus, as well as their own swine influenza viruses. And two, let's say bird and human influenza infect the same cell in pig, this segment can rearrange, they can reassort and create completely new virus, new strain, which can actually afterwards spread from pig to humans. And because it has new combination of hemagglutinin, neuraminidase, and other proteins, we do not have any immunity against this. And most pandemics are actually caused by antigenic shifts. Let's talk about pandemic. We already touched upon Spanish flu, 1918 to 1920 influenza pandemic. Now here, for example, in this source, it claims it was from 50 to 100 million fatalities, which is a little bit over exaggeration. Average estimate is from 20 to 40 million. Now, since it had such a huge impact, it was so highly pathogenic, it was for a long time considered that it was caused a virus, it was H1N1 virus, that it was antigenic shift. However, reconstruction of the virus confirmed that it actually was antigenic drift, not antigenic shift. And how the virus was reconstructed. The viral RNA was, for example, isolated from Inuit women who died of the influenza flu infection, and she was preserved in permafrost. Sorry. So the RNA was isolated from there, and there was another samples from the soldier, American soldier, which was preserved in hospital lab. And based on these two samples, the scientists were able to reconstruct this pandemic H1N1 virus. And they confirmed that actually it was a result of antigenic drift. And in addition to changes in neuraminidase and hemagglutinin, there were other mutations which enable overactivation of immune system. You remember, contrary to regular influenza outbreaks, which usually are highly pathogenic for old people and children, this influenza pandemic, the Spanish flu strain, was highly pathogenic for young people between early 20s and late 30s, and it's because their immune system was highly active and it triggered its overactivation. Afterwards, in 1957 and 1968, we had new recombined strain H2N2 and H3N2, which caused both over 1 million fatalities. H1N1 reemerged again in 1977. However, it was mild. It changed. It didn't cause such a high death rate as one in 1918. And in 2009, there was expected new critical pandemic, which turned out to cost less than 300,000 lives. And it was again H1N1. Now, nowadays, what is kind of threat is highly pathogenic avian influenza A, H5N1. And it caused several outbreaks in the farms, poultry farms. It is highly pathogenic for birds. However, fortunately, it has very poor transmissibility among humans, so it was not able to trigger any epidemic or pandemic. Now, we know that every year we have a vaccination. That's the best prevention for influenza. And this vaccine are generated on based on prediction from previous flu sequences. And basically, it's computational programs which analyze sequences of all previous flu strains. And based on that, they predict highest probability for that year outcome, and the vaccine is created based on that prediction. In most cases, most time it works, and it actually creates herd immunity, protecting those which are more vulnerable. Now, once somebody had infection with flu influenza virus, there are drugs which can ameliorate symptoms such as zanamivir and oseltamivir. Oseltamivir is what we call Tamiflu. Tamiflu targets neuraminidase, so it prevents virus being released from infected flu. And you can see it's quite efficient. If you look on this graph, you see on x-axis days after starting treatment, and on y-axis the median of symptom scores. And if you see on upper um, graph, there is placebo, so symptoms are really high, but with this little triangles, the lower line represent those who were treated with Tamiflu, so it is kind of efficient, and therefore, for example, Army and certain governmental organizations stockpile Tamiflu just in case if pandemic happens, so we have enough drugs to treat the people.